Yeah, come sit. Can you guys see in the back? Okay, everyone who, wait, before we start, we have this beautiful space, and I wanted to give the people that donated this space a little bit of time, because you have to go, but thank you to Arch Lending and Himanshu for giving us the space, and tell us about it.
Thank you for coming, guys. Thank you all for coming, and thanks for putting it up. This is a lot bigger than the last one we did here a few months ago, so it's growing very quickly, and a lot of new faces that I don't know. So Arch is a crypto-backed loan provider. We're the leading provider of loans in the US. If you have Bitcoin, Ethereum, or Solana, come to us, get a loan instantly. We also have other products for people that have crypto. Our goal really is to be the next crypto-powered bank. If you have any interest, check us out, Arch Lending. And with that, I'll turn it over to Mikey to continue. Thank you.
Thank you. Okay, who brought a costume? Let's have all the costume people come up. I know. Yes. We have a couple people for sure. Yeah, we have at least two. Do we have a third? We're going to do the smallest DeSci costume contest ever. Oh. And now we're rolling backpack. Oh wait, we have three. Wait, one more. Don't hit the camera.
I got
Try not to hit the camera. Oh, four. Okay. Okay, so we have Pikachu. We have an astronaut. Yeah?
Looks like an airplane.
Yeah.
Certain.
Yeah, astronaut.
80s gym teacher.
80s gym teacher.
Corporate monkey.
Corporate monkey. And I'm a rolling backpack. Let's give everyone a round of applause. Do you guys have anything you want to say? Any words of inspiration? Any how do you get out of being a corporate monkey?
No.
Okay, all right. Well, let's give them a round of applause. Okay. What else do we want to do? Okay, so this is a couple people. My name's Michael. I'm hosting the event tonight, and I'm hosting the event tonight along with Bryan. And we'd also like to thank Bryan, and Coinbase was also kind enough to sponsor it. Do we have someone from Coinbase here? Yeah. Luca? Okay. Well, Coinbase also was kind enough to sponsor it, provide a lot of pizza and other things. So thank you to Base. So this is a monthly science meetup. I was looking at I moved to New York a bit ago, and I thought, there's all these run clubs, and there are all these drinking clubs, but there's not a lot of things for people who care about science, or there's some tech stuff, but there's no sort of science stuff. So I thought it'd be fun to start a science meetup. So we do these like once a month for people that are interested. We have over like 5,000 people who've attended at least some of the events. We've done like 18 events so far. We put all our videos online. So if you go to DeSci.NYC, you'll find this one, as well as all the other topics that we've done. If you would be kind enough to follow us on Instagram, that would be amazing. If there's a picture from tonight that you're interested and you want to tag us with, that is always, so take out your phone. Nobody taking out their phone.
We already follow you.
You already follow me?
Yes.
I'm gonna have.
What about on Mastodon? We're a science crowd.
What was it?
Are you on Mastodon? We're a science
We're
crowd.
not, but we should because we were banned on Twitter. So.
Wait, what?
Does anyone here work at Twitter? Or know someone that works at Twitter that can unban our account? I'm not even joking. We were seriously banned.
What happened?
What did you do?
We were tweeting about ketamine too much.
Oh my
So it is actually banned. So we're trying to unfuck it. Okay, so actually tag us on Instagram. We appreciate it. We've had a lot of really cool speakers before. We've had things on mushrooms, bees, Martin Shkreli, NFTs, Research Hub, longevity, doing the Olympics but with steroids, bioelectricity, don't die, and the last one was on ketamine. So now I'm going to talk a little bit about the up-and-coming ones. Does anyone actually know who Elizabeth Fick is? Interesting. Okay. Did anyone hear about the Stanford kerfuffle where they fired the president? So she was sort of like she's a person that does like scientific integrity, and she was like integral into how he was fired and in discovering the improper stuff in his research paper. And so she's a really, really interesting person. She used to work at a company called Ubiome, which had a little issue, and then, does anyone know what Ubiome is?
Yeah.
It's a fecal with
Yeah, exactly. But she got out before then and then started this whole consultancy around scientific integrity. And it's super, super interesting. And she just won a really big award. And so I reached out to her, and she's going to be giving the talk on November 19th. If you want to RSVP, you can do so here. Then we also have Sheridan. Some people here know Sheridan. He's gonna be giving a talk on sexual optimization, and he's trying to be like the Bryan Johnson of the penis and optimize it. He's a really cool guy. You'll like him. And then lastly, we have Noor. She's doing this thing called Orchid Health, and this is around doing like full genetic testing on embryos, so you can make a life less suffering. A couple people have asked, like, what is decentralized science? And it's a sort of a complicated thing, but I think we're really hitting the stride. Like this is the year of decentralized science. It's like Bitcoin 2015-ish. Like it still needs a little time to like cook and stuff, but like it's happening. So what is decentralized science? It's how to use the blockchain to improve science. So if you think about like what are some of the problems with science today, is like there's like a weird incentives around like who gets paid for what or who gets credit for what. And what's cool about the blockchain is that like this fixes it, because like all data that you upload can be attributed to a certain person or a specific research scientist or a certain instrument or whatever it is. And the blockchain also sort of enables it, like sort of what happened with decentralized finance. So in decentralized finance, like it opened up the economic world to lots and lots more people, like sort of what Arch Lending is talking about. And what this tries to do is open up science to more people, or even more types of bots. So like imagine like not just humans are scientists, but like AI bots can be scientists, and like the blockchain can be a way for them to communicate with each other. So like you have this infrastructure for humans and bots and people to upload data and verify where it came from and get attributed to it, so that it works out better. And so I don't think this is far off. It's been around. The concept of DeSci has been around for a few years. We started this about a year ago, and I think it's starting to happen. There's becoming more and more projects that are becoming sort of just on the cusp of being successful. We're launching a token on Base shortly. So each time, if you didn't check in, make sure to find Brian or Isaiah or Jingping. Are you guys? Raise your hand. Yeah. So if you haven't checked in, make sure to check in with one of them. And what we're doing is we're seeing who is checking in and attending the events, and using this as like an airdrop for the tokens. And we sent out a couple emails to test the waters. So if you haven't gotten an email yet, just wait a little bit, and you'll be getting an email shortly telling you how many DeSci tokens you have. And so it's going to be launched on Base. We might provide a little liquidity, and then you'll be able to trade it and use it to participate in research studies, or use it for like betting and gambling on research projects, which is something we're going to be launching soon as well. So, but what's cool about doing this is like it's like a fair airdrop doing it this way. So by attending the events, you're sort of showing your commitment. You're not just trying to like farm it too much. So that's happening imminently. It'll be on base and it'll be very cool. And we have a leaderboard also of all the people, so you can go to svn.haus and click on DeSci and see who's the leader, who has the most tokens, and it's actually kind of fun. It's like gamifying science. Okay, so I started a company called SVN, and how many people here are familiar with like Polymarket? Okay, now imagine there was like a Polymarket for science, so you could like bet on like clinical trials or like which drugs are going to be passed or like what the weather's going to be like tomorrow or like all these different types of things that like are important. And what's cool about this is like prediction markets are an interesting way of allocating resources. So maybe if you have like a clinical trial and it's like very lopsided, you might not even have to do that clinical trial. So we've been experimenting with this for a couple months. So you can go to svn.haus and like bet on clinical trials, which ones are going to be passed. And this is sort of totally separate from like the stock market. So you can use that information to inform other types of markets that would be influenced by that. But I think just sort of this concept of prediction markets for science is going to be like a really big idea. So if you're interested, you can check it out. Oh, I didn't put the link. That was dumb. But yeah, so prediction markets for science. And the reason I mention it is because we're hiring. We are really sort of looking for like another developer to join us. We have a team right now, but we're looking to grow it. So if you're looking for a job or you know someone that is looking for a job and that finds like a Polymarket for science interesting, if you're in science, we're looking for like a marketing person, we're looking for a developer primarily. Like those are the two spots we're looking for, but we can hire. We're backed by Polychain, which is one of the top crypto VCs. So we're sort of very reputable, backed by Balaji, backed by Avalanche, and backed by DAO5. These are sort of the top crypto VCs. So if you're interested in joining, scan that link and apply for one of the positions. Definitely doing it through the website is the best way. If you send me a message directly, it's not as good. So apply there. And lastly, does anyone have any questions? Yes.
Okay, so this is an interesting idea, right? That a lot of these like biotech penny stocks are kind of the same thing, right? They're binary bets on clinical trials.
Yeah.
So how is this different?
Well, I mean, prediction markets work well for binary things like the presidential election, right? Like, so as you go up and up and up until the point at which it's binary, more and more information comes in and allows you to trade on that. So if for a penny stock for like a clinical trial, for those types of things, oftentimes if a drug is approved or not approved, sometimes the stock can do random things also. So what's cool about this is that it segments out the actual thing that you sort of care about and leaves the other stuff, because there could all sorts of hocus pocus that goes on within the stock. But this is different because it's just focused on the actual trial. Yeah. And down the road, we're going to be working more with clinical research organizations to actually do our own science, so you can bet on outcomes. So this is like a much different way of doing academic research. Like right now, peer review is so broken because like, you're giving it to someone, they have no skin in the game to do it. And what's cool about this is now everyone is a peer reviewer. So you're democratizing peer review and you're incentivizing it too. So now you're actually like, oh, I'm going to do this research. I'm going to check each data point. I'm going to develop a bot that's going to check each data point so that I know this is accurate. And the goal here is to create a sort of a marketplace for accurate science and accurate information so you can build on top of it a lot more. Yeah. Anyone else have a question? Yeah.
What does SVN stand for?
Science Validation Network. Yeah. So you can science, and if you pronounce it, it's kind of cool. It's sovereign, SVN, sovereign. So you can say SVN or sovereign validation network. Yeah. One more. Anyone about DeSci or? Oh, we're looking for speakers. We're gonna start democratizing speakers. If you have good ideas, if you're doing like, we're looking for speakers that are sort of crazy, like in the mix between like sci-fi and like traditional research. So half the people should love them and half the people should hate them. Any last one? Okay. So without further ado, we will bring Ryan onto the stage. And can you bring your laptop? Yeah. Oh. Okay. Yeah, that's fine. Cool. So Ryan is from Minicircles. So Minicircle is this really interesting company that is doing a reversible gene therapy. So one of the issues that gene therapies had before is if you did them and you put them in your body, you're sort of screwed, right? There's no way of doing it, getting it out. But they came up with this way of, this three way of stopping the gene therapy from growing. You're taking a chemical, cutting it out physically, or there was a third way that I'm blanking on at the moment. And so this is like a very innovative way. They sort of started with that, and you can do all sorts of these things to edit your cells. So if someone has a genetic disorder, maybe they can change it, which is a very, very new way of doing science. So I'll let him explain a little bit more about how it works. But yeah, come on up. And they're doing all this interesting research. They're doing both research, and they're doing also clinical work in Prospera. So there's this place in Honduras, I think, that you can actually, because you can't get it done here, as far as I know. Maybe there's some places that can. But they're doing, you can fly to Honduras and get these gene therapies done, which is very cool. And Bryan Johnson recently did this for this thing called follistatin. So now he's like super buff. And he's not doing it just to become buff, he's doing it to live longer. Because a lot of people, you know, as they age, they sort of fall downstairs and... Good? Thank you. Okay. All right. Boom. All right. Looks good. So, without further ado, let's give Ryan a round of applause.
All right. Thanks, everybody. I was happy to hear Michael say if you're looking for speakers who are a little bit crazy. I'm definitely a little bit crazy. Have a crazy job. I thought there was going to be like 5 to 10 people here tonight, so it's a totally different audience than I expected, but I'm excited to talk to y'all. Hopefully some of the stuff I share, you know, piques some of your interest. Yeah, so let's get into it. I'm not very tech savvy. I feel like I have to push the arrow to move it. That's just the way it works, right? Okay. Okay, so. So, yes, my name's Ryan. I work for a company called Minicircle in Austin, Texas. Yeah, so let's see here. So just an overview of what I'm going to talk about tonight. I heard I have about 30 minutes. It should be no problem. So basically 3 parts to this talk. First will just be a little bit of an orientation to aging and longevity science. I'm sure some of you know a lot about this. I suspect maybe some of you don't. My PhD is in molecular biology of aging, so I like to just offer some background there. Then I'm going to talk about the specific work we do at Minicircle. As Michael mentioned, we make a reversible gene therapy, which is novel. It's also a plasmid gene therapy, which is something I'll get into. It's unique or different in a few ways. Yeah, so, and then at the end I'll conclude with some just some thoughts on biohacking longevity. It's something I think about a lot. Again, as Michael mentioned, Bryan Johnson's one of our patients or collaborators. So yeah, I'll talk about some stuff like that. So first of all, a little bit about me. I'm definitely a little bit crazy. I have a very colorful background. So I was an NFL athlete and a college track athlete. I feel like this is relevant to the science of longevity because people like Bryan Johnson are basically professional longevity athletes. I'll talk about that at the end, but there's really a performance aspect to this, and there's things that athletes do really well that scientists and maybe some people in the longevity space aren't as good at. Science, I'm kind of an outsider in science. I came to science in my late 20s. I'm in my early 40s now. I just got hooked on science, moved to Seattle, got a PhD in the molecular biology of aging. Did a really cool postdoc with the Air Force on a DARPA project called biostasis, which was basically drug-induced human hibernation. It's almost certainly possible. Really, really interesting stuff, super fun. And after that, I was extremely burned out, and I bought a van, lived in the deserts and the mountains for a while, taught 6th grade science, and now I work at Minicircle in Austin as the director of longevity research. So things are good. The third thing on here is communication. So I feel like generally science has a bit of a communication deficiency. A lot of the most exciting and fun things about science are not communicated to the public, and sometimes concepts aren't made simple enough. I'm a big believer that it's the simpler you can explain a complicated concept, the better. There's nothing good about making it complicated and long-winded. Just get to the point, give people actionable stuff. Really, really into that. So, so the way this is kind of the way I think about it. I think these three things, science, communication, and performance, can kind of converge to advance longevity biohacking, and that's kind of my goal tonight, to touch on that as well as the specific work we do at Minicircle. So, just a little bit of background about the history of aging research. Sometimes I kind of prefer to call it longevity research. The term anti-aging is really not a popular term in the aging research community, but we've studied what we're really trying to do is enhance longevity at this point. So the way I tell this story to people is, in 1935 we starved some lab rats, and they lived longer, and nobody knew that was possible. And then for 50 years, the field was like, how does that work? We had no molecular biology tools, nobody knew. We didn't even discover the structure of DNA until 20 years after that. So, there were these early seminal experiments, and this rate of living hypothesis was a really influential, kind of theory. But in the interim time after this initial, dietary restriction experiment, I feel like there were a lot of theories developed. Denham-Harman's mitochondrial free radical theory of aging in the 50s, very intuitive theory about, just oxidative metabolism is what slowly kills you. Let's see, Hayflick limit was established, disposable soma theory, antagonistic pleiotropy. From 1988, or most people would say 1993 probably, through 2004, this is like the inflection point for the history of aging research. So, does anybody know the specific experiment I'm referring to in 1993? Okay, so Cynthia Kenyon at UCSF mutated one base pair in the worm insulin receptor. It's called, the gene is called DAF2 in worms. She doubled the worm lifespan. So I think worms have like maybe 10 million base pairs. Ultimate butterfly effect, one base pair mutated, doubling of lifespan. So what this led to is a bunch of whole genome screens in model organisms like yeast, worms, flies, and some mouse work as well. Basically what happened here is we kind of, fleshed out this holy trinity of fasting activated longevity pathways. I would say it's insulin gets turned down, mTOR gets turned down, and AMPK gets turned up. That's a really powerful paradigm. There's other threads in longevity that are parallel to that, like telomeres and senescence and all kind of things, but this is really important. After that, there was a lot of expansion of the field. So we looked at, like, if you turn mTOR down or insulin down, what does that affect? It affects hundreds of genes, and we define those through further genetic screens. Yamanaka discovered the Yamanaka factors. So when an egg is fertilized, an egg is an old cell. Eggs are like 20 plus years old. necessarily, or ballpark. I mean, but I think it's day 5 after it's fertilized, the Yamanaka factors go in and completely reset the biological age of the fertilized egg. And so now a ton of money, billions of dollars are going into applying these rejuvenation factors to basically adult humans. This is called partial reprogramming. Maybe the hottest thing right now. Yeah, Yamanaka discovered that. There was a parabiosis experiment. They sewed an old mouse and a young mouse together via their circulatory system. Young mouse got older, old mouse got younger. So proof of principle of vampirism. Perhaps disturbingly. I mean, it is what it is. Senolytics, Aubrey de Grey's sends. 2013 through 2020, the field aging is a field kind of like psychedelics with a history of fringe associations. Struggles to get credibility. 2013, we really got mainstream traction. So Google, or Calico technically was the first, I think, to put a few billion into Calico. The Hallmarks of Aging paper came out in 2013. This is kind of the Bible of aging research. The geroscience paradigm was developed. That basically says, hey, NIH, you're treating the major killer diseases, atherosclerosis, cancer. If you treat aging itself, you treat all of them at once. So, because aging is not a disease, so it can't be an FDA endpoint. So, very frustrating bureaucracy at times. The FDA does a ton of good stuff, but the TAME trial, basically the idea was type 2 diabetics taking metformin were living longer than non-diabetics was the idea. So it's like, hey, can we give metformin to non-diabetic people? Maybe give them some of the benefits. So on and so forth. This I added recently. I mean, things are really, I think, getting crazy now. Hevolution is the Saudi royals put maybe tens of billions of dollars into aging. Altos, the Chinese He Jiankui thing, AI, Bryan Johnson's blueprint, rejuvenation Olympics, gene therapy. Science fiction is becoming reality in a very public way at this point. It's happening very fast, and it can be a very good thing. So, intro reading, I always recommend this to people who are new to the field. Hallmarks of Aging paper. This book, Ageless by Andrew Steele, is probably the best intro book, so for what that's worth. Human aging and death. Cardiovascular disease kills more humans than anything else. It's also one of the easiest things to prevent. Avoid cardiovascular disease. Cancer and neurodegeneration are more insidious, harder to stop, in my opinion. Frailty, stem cell exhaustion are also very real. You won't, I think if you look past 70 years old, you won't see a lot of people who can pass for really, really youthful. So this is something that's always good to keep in mind. Some people in Austin asked me for the 80-20 of longevity. So I would say if you really want to be good at longevity, avoid catastrophe. Don't shoot yourself in the foot, okay? So, Michael was talking about betting earlier, make safe bets, okay? Don't smoke, that's a safe bet, stuff like that. Do the common sense good things. Sleep, eat healthy food. I would say Mediterranean, maybe with occasional low carb and fasting, is a very safe bet. People like all kind of that, but that's a very safe bet, I think. All the good things, everybody knows this stuff. Aggressive self-data collection, I would really encourage. So, I'll talk about this later, but whole genome sequencing, it's $500. Raise your hand if you have your whole genome sequenced.
That's 23andMe.
No, great question, thank you. And please, anybody yell at me, ask questions, whatever. 23andMe sequences less than 1/1000th of your 3 billion base pairs of DNA. You can get all 3 billion base pairs sequenced 30 times over to eliminate any uncertainty for $500. And nobody knows this and nobody has it done. People say stuff like, oh, I wouldn't know what to do with it. Just get it. You can spend the rest of your life figuring out what to do with it. You can get your whole genome sequence. That is what makes you you. That is the base layer of your biology. Get that data. Anyway.
How do you get that data?
What's that?
How do you get that data?
Sequencing.com.
Thank you for answering this.
Prometheus, I think, is something you use to process the data. I don't. Sequencing.com, our CEO did it through Nebula. There's like 3 Sequencing, Nebula, Dante, I think. I'm not affiliated with these companies, but just, yeah, I mean, it's $500. It's great, you know, spread the word. And then, so don't do the bad things, do the good things, and consider advanced stuff, okay? So you gotta be careful here. I mean, gene therapy. Before I worked for Minicircle, I volunteered for the clinical trial on an island off the coast of Honduras. I was like, this might be the last bad decision I ever make. I had no idea what was gonna happen. It was way better even than I hoped, but you know, it can get a little dicey out there. Historically, I'm a pretty risk-accepting person. I'm not evangelical about that. It's definitely backfired quite a few times. But yeah, so acquire information and access around cutting-edge stuff. So read, network, Rappa, metformin, that's mTOR and AMPK. ICAR is like potentially a holy grail drug that nobody knows about. Just remember that word, ICAR, okay? Google it, trust me. Senolytics, beyond that, you know, gene therapy, stem cells, blood-based therapies. Way beyond that, maybe biological replacement. That's actually a thing now. You know, cyborgs, AI integration. Love him or hate him, Elon said, I heard him say the human brain is a bandwidth problem. I agree strongly with that. Like, I'm basically like a brain athlete. I have to read a bunch of papers. My brain is not up to the task. Like, it's just getting by, like, you know. So anyway, this is some stuff about longevity. It's another way to think about it. You know, you have the base of your pyramid. No drug can fix a terrible lifestyle. You got to have the foundation there, and then you can work your way up the pyramid. Okay, so that wraps up part 1. You know, a little context about longevity. Now we're going to talk about the specific stuff we do at Minicircle, gene therapy company. Yeah, what's up?
Quick question.
Yeah.
What do you think about PI1 inhibitors
PI1 inhibitors?
for fibrotic disease? It's a signaling hub.
I don't know much about them. Yeah, I'd love to talk to you after, but I do want to say this. If you're a scientist or if you're anybody, it's really important to just be able to say, I don't know. Okay? It's super important. So yeah, I don't know. Great question.
Can you go back one?
Yeah. Yeah, and if anybody likes anything on these slides, I can email you the whole slide deck, whatever, take pictures, it's all good. You know, just holler at me. I'm super easy to get in touch with. This is all I do. 24 hours a day, this is all I ever do. I'm not married, I don't have kids. Like, a few years ago, I crossed some threshold where it's just like perma on. Okay, this is it for me. Okay, so I never get tired of talking about this. It's all I do. So holler at me. It's all good.
Can you comment on the cyborg decoupled consciousness aspect of that thing?
Yeah, sure. Yeah. I mean, so all this stuff is physical. Okay. I think consciousness is like, we don't really understand it that well. But I don't know, if I just use my imagination, I feel like at some point, you know, you're just going to be like a cloud or something that can, I feel like decoupling consciousness from the physical container is probably beyond this like cyborg, AI integration, machine parts. It's just a hunch, yeah. So.
What do you mean by that, decoupling consciousness from physical container?
Yeah, I mean,
do you think
like
like the brain and
playing
body?
around
Do you think consciousness is separate from the brain and body, or do you think consciousness is this whole other different thing? Like,
yeah,
more
I don't
like
have a.
physicalist or like dualist.
Sorry, can you speak up?
Are you more like a physicalist or like a dualist? Like.
Yeah, I'm not trained enough in that vocabulary, but I'm kind of a common sense guy. I would say like. I've seen considerable evidence that consciousness is not limited to the physical body. I don't know what that means, how that all shakes out, but I feel like in some way, I feel like the pyramid is basically physical, as I've described it here. I think beyond the cyborg, where you start becoming biology and machine merging, that's still physical. Beyond that, I feel like you just separate from the physical. Just broad strokes here, okay? I'm not an expert in these fields. It's just a fun talk, you know? I'm just talking.
What's the considerable evidence that you've seen that consciousness is not in the body?
What's that?
You said there was considerable evidence.
Yeah, I would say that, yeah.
Can you elaborate?
To be honest with you, some things I cannot elaborate on.
All right. Fair enough.
I would say, I think we've all had experiences where like maybe dreams can be predictive, or there seems to be, you know, instantaneous, like spooky action at a distance involving consciousness. I've seen strong evidence for psychic capabilities in humans. Anybody watching this on YouTube, you can laugh at me all you want. You know, whatever. I've seen enough that like it's a very weird world that we live in. Okay, people have known this for a long time. I hate stuff being stigmatized. Okay, it's the enemy of knowledge. I hate it. You can probably hear it in my voice. I hate it so much. We should be free to talk about stuff. Okay, it's the only way knowledge. We're a spacefaring civilization. Okay, we have to act like it. Okay, no more stupidity, stigmatizing stuff. Gotta get past that. Okay, so yeah. Okay, good crap. All right, so yeah. Speaking of weird stuff, history of DNA and gene therapy. These two guys. Yeah. What's that?
Watson and Crick.
Watson and Crick. Yeah. Yeah.
And Rossner.
And Rosalind Franklin. I wasn't there. I don't know the whole story. Watson and Crick are so iconic. If they did steal stuff from her, that's messed. So I don't know what happened, but I put her in there, and that's the original. I think that's the first sketch any human ever made of the DNA double helix, more or less. So yeah, this is like really like human history stuff. I mean, it's so iconic. You know, speaking of weird stuff, there are rumors that these guys were on LSD when they discovered rumors. And then Crick definitely went on to write a paper in the 70s called Directed Panspermia, in which he speculated that life on Earth could have been seeded from outer space. Okay, so you can't get much more credible than Francis Crick. He totally wrote that paper. It's available. It's a very weird world. Okay, so and science is not at all incompatible with weird stuff. So 1950s, we finally described the structure of DNA. 50 years later, we kind of sequenced the human genome, and then right at the same time, you know, some gene therapy, viral vector trials, and like there were child deaths, and this was a very big blow to the field. You know, it's a tragedy for the children and the families involved, and it was so many articles were written in the wake of this. So that's an important point in the history of gene therapy. Gene therapy is dangerous, so you got to be careful. 2010s, you know, CRISPR-Cas9, partial DNA sequencing becomes mainstream. You know, you get like they cherry pick like under 1,000th of your genome. They're like, okay, here you go. And then apparently maybe later they sell your data or something. I just read the headlines, I don't know. But Liz Parrish, she's our collaborator. We talk to her occasionally. I've never talked to her, her CEO does. She went down to Columbia and got follistatin gene therapy. You know, my mentor commented on it publicly. It was a thing. 2020s, I guess one of the COVID vaccines was viral gene therapy. Whole genome sequencing becomes affordable. No one knows about it, okay? Get your genome sequence, seriously. Bijan Kui, you know, genetically modifying embryos, babies, or whatever. And gene therapy startups popping up, including us. So, Minicircle, this is us. So, reversible plasmid gene therapy. These are our two founders, Mac and Walter. These guys are like 30 years old. I wouldn't be here. I get to travel all over the world, meeting people, talking about science on a company credit card because of these guys, okay? I'm like a hired gun at Minicircle. But these guys, they've never had real jobs. You know, they're 30 years old. They're extremely unconventional people, but they're both brilliant. I've never built a company. They built a company. They're good guys. I vouch for them. So that's them. in their mid-20s, they spent like 5 years doing R&D to develop a plasmid gene therapy, which I would describe as an irrationally neglected mode of gene therapy. So the whole field kind of gravitated towards, I guess, viral vectors. Plasmid, there's no virus involved. It's just some DNA. People say plasmids don't work, but they are generally thought to be pretty safe. There's a series of papers by a guy named Mark Kay at Stanford that really dug into plasmids. A lot of stuff is, developed on that. So basically with our system, instead of like IVing a virus carrying a genetic payload, what we do is we just inject into belly fat and transform a few belly fat cells, and that basically sets up a little export factory of, in the case of our gene therapy right now, follistatin. Okay, so this is basically complementary to viral. So it's probably less powerful. It's also safer. It's about an order of magnitude more affordable. Right now we're limited to secretory proteins. So some proteins cells make with a little secretion tag, they get exported to the cell surface, sent into circulation. We can only use those proteins with our plasmid system right now because we're just transforming a few cells. But we're working on it, and multiple layers of reversibility. So tetracycline is the first and most elegant layer. That's continuously under refinement. And then next would be a local injection to dissolve the fat cells. And then third would be you just excise the fat cells. So multiple layers of reversibility. Like I said, it's a very safe mode of gene therapy to begin with, but you want to err on the side of safety when you're doing radical stuff like this, okay? Yeah?
Sorry, can you talk a little bit more about how the plasmids get localized to a specific area and stay in that area?
Yeah, so the plasmid is injected with a cationic polymer. So like DNA is negatively charged, won't get through a cell membrane. We mix it with a cationic polymer, which is positively charged. There's a lot of literature describing how that gets into the cell, endocytosed, gets into the nucleus. So it's kind of sticky is the word we use around the office. That positive negative interaction tends to stay pretty local, we think, and that's the thing there. Yeah?
Yeah, what happens if the cells undergo lipolysis?
Yeah, I mean, if the cells undergo lipolysis. So it's a good question. I think of lipolysis from an athlete perspective, basically. This is an open question. Does that involve breaking down the entire cell or just like triglycerides in the cell? Do you know?
Well, let's just say if you did a week-long fast and just burned off some fat.
Yeah, that's a great question. The short answer is I don't know. Okay? I mean, a lot of people who are kind of involved with this do a lot of fasting. Bryan Johnson among them, myself. I would say we have not seen anything suggestive of fasting or fat loss negating the therapy, but it's a valid question, and I don't know the full answer to it. Yeah. Okay. So yeah, this is the same talking points here, but just a little cartoon of, there's a plasmid, it's like 10,000 base pairs, injected into some belly fat, gets in a nucleus. It doesn't integrate, this is a key point, it doesn't integrate into your pre-existing DNA, it just kind of sits next to it. It's like a little teeny tiny extra chromosome. And it just makes one gene, follistatin, and that has a signal peptide on it, so it gets secreted and exported into circulation. Just a little export factory, and some of the benefits we see are increased fat-free mass, decreased body fat, and decreased epigenetic age. Yeah?
Why does it only last 2 years?
Guys, let me say one thing. Or y'all, let me say one thing. Am I hearing so-so? Please speak up if you ask questions. I heard you though. Why does it only last 2 years? I wouldn't even say it lasts 2 years, okay? I'm not a salesperson, okay? I'm a scientist. I'd say a year is maybe good, but why does it only last that long? So I think eventually, it gets silenced via methylation. Eventually. I'm not 100% sure. I've actually been digging into the literature recently to really try and understand this as deeply as possible. That's what I can say. Yeah, right there.
Yeah, you hear me? Good?
Yeah.
Okay. So have you, in which settings have you tested actually the plasmids? So have you done like mouse models or some organoids or human beings? And how did you make sure that the plasmids are still in the cells and they're not being degraded, for instance?
Yeah, it's a good question. So, I've been working at Minicircle for 1.5 years. Like I said, Walter and Mac did like 5 years of R&D before I got there. They did human work. They did a lot of cell culture work and some mouse work. I'm going to be honest, it was a lot of DIY bio. It was scrappy and spirited, but it's pretty like, that's what I would say. I think we see persistent benefits. We do a lot of ELISAs internally to measure. We have a pipeline now with a mouse and cell culture company where we can like pump volume through an experimental pipeline to replicate some of these old internal data and just flesh out that data package more rigorously. So that's about where we're at. Yeah, good question. Yeah.
I was slightly Google, does like the fat-free mass have like any benefits towards like longevity?
Yeah, that's a great question. I would say yes. Fat-free mass, probably good for longevity up to a point. So I would say FFMI, fat-free mass index. BMI is body mass index, not a great metric. Fat-free mass index is a good metric, I think. We have a lot of data from bodybuilders, and we know when steroids were made available, I think it's the mid-50s. Fat-free mass index basically maxed out at about 25 pre-steroids. Post-steroids, Ronnie Coleman, I think he's like at 40, fat-free mass index, 35, 40, something like, different world. But I think 25 is maybe a reasonable, if you're not at 25, that's, I don't think you have, but muscle frailty occurs with age. Okay, sarcopenia, so muscle weakness and muscle loss. Bone loss too. You want to have muscle in the bank going into beyond middle age. Also, muscle is a glucose sink, so that's another reason. But yeah, you want to be strong. If you want to age well, you want to be strong. You also want to be cardiovascularly fit. You want to be a lot of things, but strong is one thing that's good. We'll talk a little bit more about that later. Yeah.
If the goal is to increase, the concentration of these secretory proteins, like what's the advantage of doing it as a gene therapy as opposed to mRNA or injecting these biologics?
What's the last part?
What's the advantage of doing it as a gene therapy as opposed to
mRNA or what?
RNA-based or biologics correctly?
What's a biologic?
Like antibodies, proteins.
Yeah. So I think just that the half-life. So mRNA is really a temporary thing, if I understand correctly. It's a temporary intermediate. So in the whole COVID discussion, I just want to say that one of the first things they teach you in biology, central dogma of molecular biology, DNA leads to RNA, leads to protein. Those are pretty much one-way arrows. mRNA is like an intermediate, okay? It doesn't go back and like massively mess with your DNA. It's just like a messenger. That's what the M stands for in mRNA. But yeah, if you inject growth hormone, it lasts like a day or something. If you inject a plasmid that makes your body secrete growth hormone for a year, it's 1 injection a year versus 365 injections a year. So that's the main reason. Does that answer your question?
Yep.
Okay. All right. Love the questions. Keep them coming. Michael.
Yeah.
Getting a lot of chit chat here. If
Fine, just continue.
Good. Okay. Cool. Okay. So speaking of muscles, follistatin, our first target gene. That is a mouse with quadruple. That's a flayed mouse, I guess, with quadruple musculature. Okay. So follistatin really, really works in animal models. We do not see that in people. Okay. People do not develop quadruple musculature from our gene therapy. But in science, science is hard, okay? People build careers on like and people spend tens of millions, on like 5% marginal improvements. You want to see big effect sizes, and you also want to see safety. So follistatin has a great track record of big effect sizes and safety. It does something desirable, it improves body composition, and it's very likely rejuvenating at the cellular level. So that kind of adds up to, we're doing something that's very intrinsically radical. We want to grab low-hanging, safe fruit at first to just get the foot in the door. You don't want to go double crazy. You know?
That like using the research that we've done on like increasing the size of like chickens? And, like,
What about it?
Like, is it like the research that we have on making our poultry bigger?
I'm sure they do. My impression is they do all kind of weird things with poultry. I don't know what all they do. Wouldn't be surprised. I'll show you on the next slide. I'll touch on that a little bit. Short answer is I don't know. Good question though. So yeah, big effect size, safe. This is an important, slightly technical point. People say it's a myostatin inhibitor. It inhibits myostatin and activin. That's more for some of the scientists in the crowd, but increased muscle mass, decreased inflammation. I think the activin is likely responsible for the decreased inflammation. Anyway, people like pictures, more big muscles from follistatin, okay? So we have a cow, not a chicken, but also an animal we eat, a cow, with a crap ton of muscle. And it's some dog with also a lot of muscle. What's that? I can't hear you. No, I don't. It might be. I don't know. I don't think it is. I didn't like vet it deeply, but I don't think it is. Good question. So yeah. So follistatin overexpression or myostatin inhibition, they can really work. Okay. So our science, we did. Early on, I think we got money from big tech people, and they were like, "Look, you got two choices. You can go the traditional route through the FDA, or you can go decentralized." And it's like, okay, let's go decentralized. So we did that. We have a network of like biohackers and high net worth individuals. That's a Venn diagram with a lot of overlap. But there's these people wanted to be in this trial. We weren't like going to an island off the coast of Honduras and like tricking locals into participating in some dangerous trial. These are people who really, really wanted to be in this trial. Okay, I was one of the people before I worked for me, so I was in the trial. Okay, so 43 men and women total, so age 23 through 88, so pretty diverse, sex and age diverse adult human sample. Look, I've looked at a lot of data in my career. I analyzed the PDFs coming from doctors' offices, okay? I was like, oh my gosh, it works. Like, this is great, So we saw improved body composition. On average, these people did not change their lifestyle, they're not athletes. On average, we saw about 2 pounds increase in fat-free mass, about 1% decrease in body fat. There were some exceptional case studies. We saw improvement of epigenetic age estimates, and importantly, no adverse effects related to therapy. So, scrappy, spirited, but a decent result, okay?
We can find this paper.
Yeah, if you Google like Minicircle follistatin preprint, I think it'll come up. Also, if you just holler at me, I can send it to you or whatever, but it's not hard to find. It's out there. Yeah. It has not been submitted for peer review yet. I'm literally working on that as we speak when I'm not traveling. Yeah. But we are committed to doing rigorous science via peer review, etc. We're not trying to do science on YouTube or anything like that. Like, just to state that. So, via our internal ELISAs, follistatin increased over twofold. That's largely supraphysiological value. It's well tolerated, no adverse effects observed. Body composition improved by about 2 pounds on average, about 10 pounds max increase, and body fat decreased by about 1% on average. Body composition improved in all groups, so male, female, young, old. It's good when you see consistent effects across groups. Extrinsic epigenetic age decreased. So this is a methylation clock. Depending on your point of view, you could call it cutting edge or you could call it not ready for prime time. I think it's an interesting tool. You just don't want to like bet everything on it. It's new. But it's very interesting. So, and we saw a decrease. It was very clear in the data. So that's encouraging. Yeah.
What timeline is this on?
3-month observation. Yeah, good question. Yeah. So future for us, challenges and opportunities. Challenges, going decentralized, we have to impose on ourselves a high ethical standard with less external imposition. That's really important. I view, to me, longevity is really a competition against death. A very formidable opponent. It's a collaboration among scientists, okay? No matter how much money you make, you can't beat death yet, as far as I know. So like, chill out with stealing IP and stabbing each other in the back. Like, we have a big project that we're working on. Let's just get it done. That's how I think about it. Not everybody thinks so. And we're currently limited to the secretory pathway, but we're working on that too. Opportunities, innovation. There's so many target genes, even in the secretory pathway alone. We have an opportunity to bridge the gap between DIY biohacking communities and the more establishment, pharma, academia, regulatory communities. And just broader biohacking advancement, which I'll talk about in a second. Biohacking. Bryan Johnson. I really like this picture. I'm a big fan of Bryan. Nope, I'm not like a fanboy, but I talk to a lot of people about Bryan. Most people, the first thing they say is, that guy's weird. I'm like, he's done a lot for science. Pretty big fan of Bryan. So, Bryan Johnson's blueprint, I argue, and I said this to Bryan, and he was like, yes. Like, seemed very happy to hear this. He's the first person that I know of to publicly share extensive personal data and a comprehensive and digestible longevity protocol. Okay, there's many experts in the field, academics, etc. Nobody did this. Nobody put it out there and said, here's all my data, here's everything I do in a given week. People need to do that, okay? You're gonna be wrong about some stuff, it's okay. We all learn when we share information and approaches. So Bryan did that. He basically spent millions of dollars on R&D and just shared it for free. Now he's selling stuff, which it is what it is. You gotta make money, but in my opinion, he's legitimately advanced the field. Dave Pascoe and others followed suit. We got some other people's, comprehensive protocols. Seem like I perceive some convergence of strategies here and maybe a plateau, but it's good that people are sharing their protocols. We're able to talk about this and think about it. Rejuvenation Olympics is tied to this. I think this is a truly great idea. There's only one metric though, an epigenetic clock. To me, there's blind spots built into that. You could be very frail and have you could be the world champion in epigenetic clock, and you could have osteoporosis or something. Aging has a lot of components. Longevity has a lot of components. I'll talk about this in a second. And there's also no transparency clause, okay? So I watched this, I think, kind of get put together from close to the beginning, and I was like, if you don't tell people they have to disclose their entire routine and have some mechanism of at least approaching that, people are just gonna figure out a way to hack the clock, and then whatever they're selling, they're gonna be like, this is the only thing I took. I think there's some evidence that has happened. As a former professional athlete, I've seen this a million times, okay? Athletes are pathologically driven to win. And they don't want to say what they did to get there. They just want to be like, it's because of, whatever, God or my greatness, whatever. It's usually a lot of other stuff too. So I think this is great, but I think this can be improved upon. Athletes, I am obviously biased. I'm like, and in many ways a dumb jock who later worked very hard to become a scientist, but athletes are great at obsessing and doing the impossible. Humans have run nearly 200 miles in 24 hours. I feel like that sounds impossible. But ultra marathons are mainstream now. People have figured out how to do this. Strongman. So many people have deadlifted over 1,000 pounds. Raise your hand if you've deadlifted 100 pounds. 200. 300. 400. 500. Okay, 1,000 pounds. Okay, I watched Half Thor deadlift 960 a few weeks ago. Unbelievable. Okay, unbelievable. It's known, and then climbing sheer rock faces without ropes, like, I would never think that's possible. Yeah, so if we aim these type of people at longevity, I think we'll crowdsource solutions really fast. How do we score longevity? Probably not just a methylation clock. Back in the 90s, Marvel had these X-Men trading cards, and they had like these stat meters for different things. I think we should have this for longevity, basically. So, you know, you have a genetic and epigenetic component, you have like a body composition and strength component, cardiovascular, detoxification, cognitive and brain health. To me, I like round numbers. 10 categories, 10 points each, max score of 100. I did this for myself. It really helped me identify my weakness. Okay, I'm a former athlete. Like, I have enough muscle. I need to focus on like my kidneys and other stuff. So, just a thought. I think this could really help the community. My data, nobody follows my Instagram, but my data's out there, you know, for what it's worth. My routine is also out there. I think more people should do this. You know, so much, I scrape and claw to advance my knowledge and my access, and so much becomes through conversation. Should have more conversation around this.
Question.
Yeah.
What actually causes aging?
What causes aging? It's a great question. A lot of things. I would say hallmarks of aging. We'll get to that. What causes aging? I would say. What causes aging? Can you speak up?
I hear about free radicals.
Yeah, I would say one thing that causes aging is evolutionary neglect. Okay, so nature really cares about you, about getting you to reproductive maturity. And then it's just like, okay, die slowly and eventually painfully, most likely. There's no selective pressure after you reproduce. There's no maintenance evolutionarily. So that's a major thing. The mechanisms at a cellular level, yeah, the byproducts of an oxidative metabolism, I would say in the 90s, people kind of did some gotcha science and were like, oh, some oxidative bursts are necessary. I think free radical, I think it still holds up more or less, but like epigenetics is the conductor of your genetic symphony, okay? And when you're young, it's a beautiful symphony. It's amazing. When you're older, it's a cacophony. The conductor has just, he's dead, and there's just some noise. Okay, so epigenetics is a big thing. Yeah, I mean, entropy, I think. A bunch of things go wrong. Yeah, so.
Shortening of the telomeres.
Shortening of stem cell exhaustion, shortening of the telomeres, yes. Senescent cells, when you're young, you have a small percentage of senescent cells. As you get older, you generate them more quickly and get worse at clearing them. So the older you get, you're literally just like carrying around dead tissue. It's disturbing. Aging, death are often disturbing up close. But yeah.
Is it desirable to live forever?
Well, everybody has to answer that. The word I like more than forever is indefinitely. The option to die is, and the risk of dying is always there.
What if Donald Trump never wants to die?
That's a question beyond the scope of this talk.
A little bit. It's just like, is any of this really a good goal to have?
That's an important question. So my undergrad degree is actually in philosophy and poli-sci. Okay, so I think the short answer is, if you look at the story arc of humanity, okay, I feel like it's progress towards more or less increasing individual rights and technological advancement and like stewardship, okay? And to me, like space travel, not being biologically bound to just deterioration of your body and mind, to me that's where the story arc is aimed. That doesn't exist in a vacuum. There's social, environmental components to that. I think intrinsically, yes, it's a good thing to have the option to be able to live longer, but there's the devil's in the details, okay? I think people should have the option to, if they're doing it in a just, ethical way. That's what I think. But
as you get older, you lose the ability to repair. The cells
Yep.
lose the capacity to repair whatever, because we have the built-in mechanism to be able to do that.
Yeah.
And you know, decrease of these mechanisms, I mean, it is ultimately going to cause death, you know? And
Yeah.
so my question is, gene therapy, I think of it as more of a replacement of the genes, right? And the
Not necessarily. No, it can also be an augmentation. Doesn't have to be replacement. It's very creative enterprise ultimately.
Okay.
Yeah.
Fair enough. Then, you know, the technologies now that exist, the way to identify the genes and be able to identify and kind of know where to correct
Yep.
is, I think, pretty much advanced due to CRISPRs and, for example, right? But actually,
Yeah.
I think it's the replacing with the right nucleotide, for example,
Yep.
or pasting. I think it's still lacking. The technology's not there.
It's getting there. It's all.
I know, but it's going to take too long to be able to really make this kind of things smoother.
Respectfully disagree. You know, I think that's a bit of a fatalistic argument. We're constantly advancing. I mean, people thought we'd never fly. You know, we tend to, we make progress, you know, steadily. I think it's a tractable problem. And I think we're getting there. Personally, I'm cautiously optimistic, but you know, time will tell. None of us can predict the future. We will see. Yeah.
So myostatin inhibitors,
Yep.
they increase muscle mass, but they don't necessarily maintain the quality of the muscle itself.
Okay.
So how are you, within kind of myostatin inhibitors, retaining the actual quality of the muscle?
That is not a question I've thought about very much. What do you mean by quality of muscle?
Right, because the muscle itself can be big,
Yep.
but that doesn't necessarily that the quality of the muscle with like the quality
muscles functioning.
Like the
exactly
fun-
the
the
functioning
function?
of
Yeah,
it
yeah.
exactly. It
Yeah.
might not be retained with myostatin.
Sure, yeah, yeah. I think so this is like, you know, bodybuilders are not as strong as powerlifters, even though they're bigger sometimes. There's, you know, I think generally more muscle is good, and it tends to have some functionality. We should have tested functional metrics. We did not. We will in the future. We're doing the best we can. It's an important point. Thank you. Yeah.
Is the trial still going?
Yep.
Sorry, is the trial still going?
No, that trial is concluded.
So 3 months and concluded.
We've continued to collect data beyond that, but the IRB was through 3 months,
Mm-hmm.
and that wrapped up quite a while ago. Yeah. Yeah.
So if follistatin activates mTOR, wouldn't that also increase the speed of aging?
Great question. Yeah, aging is not black and white. Okay, so things like sex hormones, growth hormones, mTOR, sometimes they're good, sometimes they're bad. You know, if you never have any mTOR, testosterone, or growth hormone, you're probably going to be at risk for frailty. If you have too much, you're at risk for cancer, other things like this. It's just about balance. You know, this debate is constantly, should you eat high protein or should you do protein restriction? It's an open question, but I feel like, you know, you want to be strong. You also want to sometimes suppress those growth signals to get those benefits. That's about what I would say.
Right.
It's a good question.
Let's finish the presentation and we can
So-
go to questions.
Summary, well, y'all already heard this. Exciting time for longevity. At Minicircle, we're working to innovate, develop a safer, more accessible form of gene therapy. Longevity biohacking can benefit from an athletic approach and more collaboration. Thank you very much for listening. It's been really fun talking to y'all.
Let's do some rapid fire questions. Let's do like, who has questions? Let's just get a show of hands. Just get an approximate number here. Okay. What? All right, go. Just go.
Do you take rapamycin and metformin?
Yes.
Okay, next question.
What would you suggest for someone who's looking to get into science later in their life like you did?
It depends on your situation, but PhD is effing grueling, okay? You know, talk to people and start reading and look for opportunities to get involved in a lab, probably at an academic research institution. It's just step by step. Yeah, just take the first step and see how it goes. Yeah, anybody? What's that?
We know testosterone ages you.
No. Hold on. No. It's not black and white. Okay, testosterone has some mini pro-aging effects, but testosterone deficiency also has pro-aging effects, such as frailty. It's not black and white. But go ahead.
Oh, but there have been many studies with castration.
Yes.
And they do live longer.
So there are data consistent with that model.
Guys, quiet.
This is one of the most debated. There's so many things in aging that are double-edged swords, okay? I would say you don't want to pick a side, okay? You got to ride the fence when it comes to longevity, okay? You need both options. Don't castrate yourself. Don't take steroids forever. You want balance. That's my personal working conclusion. Yeah.
Is it critical that the plasmid not integrate with host cell DNA, and is the company looking at ways to protect the plasmid from that integration? Cancer research.
Yeah, it's important that it does not, and I think prior literature, it's a very low frequency event based on other groups' work, but it is undesirable, and it is important that we keep an eye on that. We don't want that happening. It's a good question. Yeah.
Why plasmid? Why not mRNA?
mRNA is very short-lived. Plasmid is longer lasting. That's why.
But it's not in the nucleus.
What?
It's not in the nucleus.
What's not in the nucleus?
That plasmid.
It is in the nucleus. What?
Mitochondria.
I can't hear you.
In the mitochondria, not in the nucleus.
What? The plasmid goes into the nucleus.
All right, next question. All right, go ahead. Yeah, Red. Yeah.
What makes a forest a forest?
Okay, next question. Does anyone else have questions?
Here?
Oh yeah, go ahead.
I saw there were like some intelligence tests or measures your brain metric.
Yep.
How does that like inhibit aging?
The brain, you don't get new brain cells, and there's nowhere to go but down in many ways, okay? The brain and cognition are probably the most important things to preserve. So test your cognition regularly, seek to improve it, seek to preserve it. That's what I would say.
Yeah, go ahead.
Oh yeah, I just wanted to ask, is there, so after putting all this effort into this, do you feel like there's like a certain ideal implementation for all the work that you've done in, like, I guess, curing all this data?
What do you mean?
Like an ideal implementation, I guess, like, would you say, what was your goal for all this research that you've done? I guess, what would you say would be the best way to, I guess, implement the range of data you've collected?
Yeah, I think, generally all humans have a death sentence. So I think one big goal is just, as touched on earlier, giving people options to live longer in good health. Obviously, the founders also wanted to make money. I think that was part of their implementation. But it's pretty balanced culture at Minicircle, just kind of both things. Yeah. Anybody?
Yeah.
Go ahead.
So you had your whole genome sequence. Like, how are you analyzing it? And like, what insights are you gaining?
Yeah, that's a great question. So, as many as I can, and I'm going to analyze it for the rest of my life. Okay, so if you look at I don't want to name any names, but intelligence communities, okay, they're not like, what are we going to do with all this data? They're like, let's just get all the data. We'll figure out what to do with it. We'll analyze it. You want the data. You can always figure out what to do with it. If you don't have it, you can't do anything because you don't have it. That's what I would say. It's hard to analyze whole genomic data, but
Is there any companies that are doing the whole analysis? I mean, there are, but
not much.
not
I mean, so
the consumer side?
I'm personally, this is like a side project, personally working on it. It's hard,
Yeah.
but it's a worthwhile. You just gotta dig in and get after it. That's all. Yeah.
Are you not using AI?
That's what
for your analysis?
Well, I'm not that. I'm not that smart at everything. I'm not very good at AI. I'm doing my best. I'm working on it. Yeah.
Anyone? Okay, one more. Yeah, go ahead.
Yeah, if someone's taking an mTOR inhibitor like rapamycin,
Yeah.
would it blunt the effect of follistatin?
Great question. I don't think so. No, I took the follistatin and rapamycin. I still perceive the benefit from the follistatin. Yeah.
Okay, well, let's all give Ryan a big round of applause. And thank you all for coming, and you're welcome to mix and mingle and chill with each other for a little bit longer. So
Thanks,
enjoy.
y'all.
We have plenty of pizza in the back, so enjoy.
My friend.
I have not touched that yet.
Those are fucking sick. I should have like that.